Today, we'll be exploring multiple sclerosis with Professor Trevor Kilpatrick. I am Professor Anne-Louise Ponsonby. I'm head of neuroepidemiology here at the Florey. The Florey is the largest brain research group in the Southern Hemisphere. We are in the top 10 of the world's best brain research centres. Our work is critical as diseases of the brain and mind affect all of us in some way. In fact, every year, 4.7 million Australians are diagnosed with one of the conditions we study. And so to today's lecture. We are privileged to have a 30-minute lecture followed by 15 minutes of question and answer with Professor Trevor Kilpatrick. Trevor is Professor of Neurology at the Florey Department, University of Melbourne, and the Clinical Director at the Florey. His research interests include the neurobiology of multiple sclerosis, neural precursor cell biology, and the study of genetic and environmental factors that contribute to MS, as well as the translation of basic research discoveries into the clinic. Professor Kilpatrick graduated with a Bachelor of Medicine and a Bachelor of Surgery from the University of Melbourne in 1982 and specialised in neurology. He undertook graduate studies at the University of Melbourne and obtained a Doctor of Philosophy in 1993. He then held appointments at the Salk Institute for Biological Studies in the US and the Institute of Neurology in London. He then returned to Melbourne as a Virtual Senior Medical Research Fellow at the Walter and Eliza Hall Institute for Medical Research and the head of the Multiple Sclerosis Research Unit at the Royal Melbourne Hospital before taking up his current appointments with us at the Florey. He's published widely in various journals, including Nature, Nature Genetics, and Nature Medicine. So now, with pleasure, over to you, Trevor.
Thank you very much, Anne-Louise, and good morning, everyone. As Anne-Louise indicated, my topic for discussion today is multiple sclerosis, and in particular to explore how to better treat this condition. Multiple sclerosis is the commonest neurodegenerative disease of young Caucasian adults. It's got a prevalence of about 0.5 per thousand. It's a complex condition. And in fact, some people would consider it as a group of closely allied conditions which have common presentations, meaning that we have a common umbrella under a syndrome, which we call multiple sclerosis. It's a chronic disease with, in many instances, unfortunately, eventually cumulative disability. And although there's been a revolution in the treatment of multiple sclerosis over the last 25 years, and in fact, a very significant advance in the treatment of multiple sclerosis because of that, the current therapies remain suboptimal. And I'll explain that a little bit during the talk. Most people believe that multiple sclerosis is an autoimmune condition in which the body's own immune cells target particular proteins or antigens within target tissue. And in the case of multiple sclerosis, that target tissue is in the central nervous system, that is the brain and or the spinal cord. Now, when we're dealing with a complex condition such as multiple sclerosis, it's important to come back to first principles and to discuss what we mean by multiple sclerosis from both a clinical and also a pathological perspective. And on this slide, we see some of the cardinal features of multiple sclerosis. On the left is a cross-section of a brain of a person who unfortunately died with multiple sclerosis. And this brain has been stained with a dye called Luxil Fast Blue, which stains myelin blue. Now, myelin is the insulating coat which surrounds the nerve fibers, which transmit information throughout the brain and spinal cord. And the myelin allows that transmission to be conducted much more efficiently. And what we see here, where the arrows point, is basically what we call a plaque of demyelination. In the surrounding area, the Luxel fast blue has stained the myelin appropriately, but here we see that there's a loss of myelin. And this is one of the cardinal features of the disease, indicating that we call it a demyelinating disease. These plaques of demyelination are the typical, if not the sine qua non, feature of the disease. Now, since the early 1980s, we've had availability to us a really important diagnostic tool, which has increased our armamentarium to diagnose the condition beyond clinical acumen, and that has been the advent of MR imaging. And what you see here on this MR image are these white spots deep within the brain, which actually represent these plaques as seen on the pathological specimen. And this has been a great diagnostic boon. But it is important to remember that despite its sophistication, really what the MRI is showing us is an increase in water content in these areas. So despite the diagnostic advance, we still cannot tell on MRI whether those plaques are active, whether they're burnt out, or whether there's active remyelination occurring in those plaques. And that really represents a deficiency of the imaging technique. We can diagnose, but we can't actually understand the nature of the pathology within those areas using a standard MRI alone. Now, under the microscope, there are some cardinal features of the condition. And this is the correlate to the gross pathology, where we see the area of demyelination. There's a discrete border between the area of demyelination as seen here and the so-called normal appearing white matter on the left. But we now know that there are subtle but important differences occurring in the normal appearing white matter as well. And this is the second cardinal feature. These are small veins called venules within the brain, and there's a mononuclear cell infiltrate from that vein into the parenchyma of the brain. And those mononuclear cells include what we call lymphocytes and also more non-specific cells of the innate immune cells called macrophages. And we believe that these cells are important in actually expediting the disease. But there are other very important features which are also present. For example, the cells which are responsible for myelinating the nervous system, the oligodendrocyte, also have a tendency to undergo death, as seen here, indicating that if we're going to remyelinate the brain, we have to replenish those cells from progenitor or precursor cells. And another very important feature is that over the last 20 years, we've recognized that although we call this a demyelinating disease, there's also ultimately significant pathology in those nerve wires, in the axons, as we call them, and the neurons, which are responsible for transmitting the information. So there's very significant axonal degeneration, which occurs in this condition as well. And we now believe that the degree of axonal degeneration is critical in determining the degree of disability that a person will ultimately develop. Now, this slide from Alistair Comston and Coles gives us some insights into the key clinical features of the disease as a neurologist would see it. What we have at the outset are some discrete periods or epochs of neurological disability, which come on over days to weeks, and then typically last months before remitting. And they usually remit to a significant, if not complete, extent. And this is called relapsing remitting multiple sclerosis. But in about 50 to 75% of patients who are untreated in the past, we found that these people, after between 10 to 15 years, developed what we call secondary progressive MS, where there's inexorable disability, which worsens day by day, month by month, subsequent to that onset. But that's only part of the story. And over the last 20 to 25 years, we've now understood that there are other important features which we need to consider. For example, before the first clinical presentation, we now recognize that at least a subset of people have disease activity, which is not revealed in clinical symptoms or signs before that event. And that's been determined by the fact that we've identified people who've had MRI scans for other reasons, for example, head injuries, and they've been shown to have typical white matter lesions, consistent with what I previously showed you, suggestive of multiple sclerosis, but not with them actually having clinical symptoms of it. And approximately 30% of those people will subsequently develop clinical symptoms consistent with multiple sclerosis over the ensuing 5 years. Another important feature are these yellow pigs. And what that indicates is that it's been shown that there are approximately about 10 plaques or 10 white matter lesions on MRI which develop for every clinical attack. And this means that if we just rely on clinical relapses in terms of thinking about stability or activity, we'll be selling ourselves and our patients short. We need to have much more intense surveillance of the condition via MRI to detect whether someone is having cryptic activity and the need for treatment to inverse or reverse that activity. And here's another really important aspect of the disease which you come to understand, and that is that actually the brain volume in a percentage of patients reduces from a very early phase of the disease with accumulation of that axonal loss or axonal degeneration. What this means is that the identification of secondary progressive disease is an artificial thing. We believe that this neural cell degeneration is occurring in many people from a very early phase, but is cryptic, and there's actually a reserve. And it's only when we lose that reserve that there are clinical symptoms which develop and that they get worse and worse. So really that means that we need to be detecting this degeneration at a much earlier stage and intervening at an early stage if we're going to protect people from that important aspect of the disease. Now, this slide just indicates the therapies that we have available at this point of time. And to a T, these therapies basically target the immune system, in particular the adaptive immune system within the blood, and stops the blood cells from actually targeting the brain. And I think what you can see now is that this is focusing on one important element of the disease, but the neurodegeneration is another. And so one of the critical issues for us is to really determine the extent to which inhibiting the immune system in the periphery is basically going to inhibit the neurodegeneration which is occurring. Now, we believe that to some extent they're linked, but we also believe that ultimately the neurodegeneration might get a life of its own independent of the inflammation. And so it's very important to target the neurodegeneration as well. Another really important point is the heterogeneity of the condition. And this graph just illustrates the variability which we identified when we recruited 500 people to a genetic study we undertook some years ago. And on the x-axis here, you see disease duration, how long the people had had clinical multiple sclerosis. And on the vertical axis, you see a disability scale of severity, with 0 being normality, 10 being death, and 6 being someone who could walk approximately 100 meters with a single stick. And so what you can see here is that within 5 years, a subset of people have very significant disability. Whereas for other individuals, you see that 30 years after the disease, you basically wouldn't know, you wouldn't be able to identify that they had multiple sclerosis if you saw them walking on the street. So this is a really important feature for us as clinicians. We need at an early stage to be able to predict who's tracking in one or each of those directions, because then we can invest our more potent, perhaps more risky therapies to those people who are more likely to have greater impact from the disease, and really at an early stage trying to inhibit that activity to knock it on the head, so to speak. So with that background, there are some important current needs for us, basically, and I'll talk about how we're moving towards basically meeting those needs at the Florey throughout much of the rest of the talk. The first is the ability and the need to determine who is susceptible to MS and how to prevent it. Secondly, to reverse disease activity in the newly diagnosed patient if we can. We need to have the ability to assess prognosis in newly diagnosed patients, as I've just said. We need the capacity to detect disease evolution at an early stage. We need capacity to impede the neurodegeneration which is occurring in the condition, quite separate to targeting the adaptive immune system. And ultimately, we need to be able to repair the damaged nervous system for those who have extant damage. I won't be talking so much about that element of our research today, but obviously a very important topic. Now, in terms of understanding the susceptibility to the condition, a complex condition such as multiple sclerosis, there are three general things to consider. The first is genetics, the second, environmental factors, and the third is the bad luck or stochastic factor. That is events coming together at the wrong time to actually synergize and basically make things worse. And finally, on this topic, I think we need to actually differentiate between the perspective about susceptibility to the condition, and once the condition is extant, what are the drivers of the disease? And I've clustered this in terms of autoimmunity and inflammation, and on the other hand, neurodegeneration. Now let's turn to the epidemiology of the condition. The demographics are that this condition has its onset in young adult life, generally in the third decade of life, and it affects women more than men. When I was a medical student, the ratio is about 2 to 1. Now it's of the order of 3.5 to 1. And this indicates to us that there must surely be environmental factors which are changing to change the susceptibility to the disease, and those who are more prone to get it, a very important area and avenue of research. Another important feature is that much research has indicated, and a lot of that research is from Australia, that there is actually a latitudinal gradient in the incidence and prevalence of multiple sclerosis, with it being higher in more temperate regions of the globe. And the third is the so-called 15-year rule, in which we identify that there is change in susceptibility of the disease for migrants according to the age of emigration. So that those who emigrate before the age of 15 tend to assume the risk of the country to which they emigrate, whereas those who emigrate as adults tend to take the risk from the country from which they've immigrated. And that indicates that there must be early life factors which lay down a susceptibility which is subsequently brought to bear in the third decade and beyond. Now and when I say we in Australia, and Anne-Louise and I were part of this mix, have been very much involved in this epidemiological research over a number of decades. One of the key internationally recognized studies that we and our colleagues undertook was the Autoimmune study, which has then subsequently morphed into the Long study, in which we had the aim of identifying every incident case of multiple sclerosis in 4 regions of Australia up the eastern seaboard. Firstly, in Tasmania, in the western districts of Victoria, in the Hunter Valley, and in the Brisbane area. And what you can see here is basically a very strong change in the incidence of the first demyelinating event according to latitude, consistent with previous prevalence studies. And out of Autoimmune and related studies, we and others have really identified 4 factors which appear to be really important in laying down susceptibility to the condition. The first is smoking. And in fact, that also is a severity factor. So we've identified that for people who are smoking and who can stop smoking, that they have the ability to change the subsequent course of their MS to reduce its severity. We also have identified that infectious mononucleosis is a risk factor. Most of us are exposed to the EB virus, but it's actually the timing and the clinical severity of the condition which follows that exposure, which is important in laying down susceptibility to multiple sclerosis. And obesity is another. And so in particular, are low vitamin D, and as a correlate to that, low UV exposure. And we believe that in entirety, these factors probably account for of the order of 50% of the susceptibility to that first episode of multiple sclerosis. Now, this is observational epidemiological data. And what we now wanted to go on to determine is whether, with the knowledge of these factors, we can actually change the propensity for someone to actually have subsequent disease after that first presentation. And this has led to the vitamin D MS prevention trial, or PREVANS, which we're undertaking in Australia at the moment under the auspices of MS Research Australia. And in that study, we've now enrolled well over 200 people who are who've had a single demyelinating episode and therefore at risk of further episodes of multiple sclerosis. And we've either basically given them placebo or dummy tablets or 3 different doses of vitamin D, 1,000 international units, 5,000 or 10,000 per day, in an attempt to see whether we can inhibit the subsequent course of multiple sclerosis in those individuals. And the flip side of this is the genetics. And we know that there is a genetic susceptibility to multiple sclerosis because firstly, there's an increased prevalence in first-degree relatives of the order of 20-fold over and above the general population. But in addition, we know that monozygotic twins are more susceptible to the disease in concordance than other SIBs. So if we have a propositus who has the disease and they have a monozygotic twin and they're females, basically the second twin has approximately a 30% risk of developing clinical multiple sclerosis and a 50% risk of having MRI changes consistent with MS. And we've known for over 40 years that there's a particular region of the genome called the HLA, which has particular alleles which provide susceptibility to multiple sclerosis. And this is consistent with the autoimmune hypothesis because these particular molecules called HLA are the molecules which are responsible for presenting antigen, and in this particular instance, self-antigen inappropriately to the immune cells, which then target the central nervous system. But over the last 15 years or so, we've been able to extend our understanding of the genetics of MS based on whole genome studies called GWAS, Genome-Wide Association Studies. And from these, we've now identified approximately 200 loci, which contain susceptibility genes. And this is what we call a Manhattan plot, which shows those areas of susceptibility for each of our 22 autosomes. But what you can see here as well is that the major skyscraper remains HLA, and the other susceptibility regions in isolation are only minor factors in terms of the susceptibility, and we're still trying to work out how they group together to provide a pattern in terms of linkages in terms of the overall susceptibility to the condition. Now, this slide shows some output from a study that we undertook some years ago under the umbrella of the ANSGene consortium, which Justin Rubio at the Florey is the current chair of. And this shows one of these areas of the genome with these susceptibility regions, and we call these SNPs, susceptibility nucleotide polymorphisms. And what you can see is in this zone of chromosome 12, in this particular instance, there are a whole range of SNPs which are changed. And these represent susceptibility SNPs for multiple sclerosis. And what you can see under here are some 17 genes in this zone. And this has been one of the areas of perhaps controversy, because the GWAS shows us zones of susceptibility. It doesn't show us individual genes in isolation. And it's very hard to pick out which is the susceptibility gene from this GWAS approach in isolation. And there's been a tendency for people to say, oh, this is my favorite gene here. This must surely be the susceptibility gene here in this region, which is driving susceptibility. You cannot do that. And we have to have more sophisticated approaches to dissect out which amongst these 17, in fact, is the responsible factor for driving susceptibility in that circumstance. And that's one thing that we've done in particular for one gene in one susceptibility region, which has tweaked our interest and represents a major area of research interest for us at the Florey now. And this is in particular the MERTK gene, which you've shown indisputably is a susceptibility gene for multiple sclerosis. And most interestingly, what we showed when we looked at the expression of the various genetic variants of this gene on immune cells, that there was a variable expression profile of the MERTK gene in these immune cells, with some genetic variants expressing low levels of MERTK and some allelic variants expressing high levels of MERTK. And when we combined these data with a knowledge of who expressed the major susceptibility allele, the HLA gene, which I talked about before, we found that those people who had low expression of the MERTK gene, the GG people, so to speak, had a much, much higher incidence of developing secondary progressive multiple sclerosis, as opposed to those who had higher expression of the MERTK gene on the immune cells. So this showed us that the MERTK gene was not only a susceptibility, but also a severity gene for multiple sclerosis, which very much tweaked our interest. Now, the MERTK gene is actually one of three, it encodes one of three proteins of a receptor family, which are very important in controlling autoimmune disease. I'll give you some background related to that. The first of these, called TYRO3, and these genes collectively, or receptors collectively, are known as the TAM receptors. And that's because they're named TYRO3, AXL, and MERTK. TYRO3 is expressed by the oligodendrocytes, the myelinating cells. I won't talk about that so much today, but we've got a very active research program about TYRO3. And AXL and MERTK are actually expressed by the innate immune cells, some of those cells which infiltrate the brain, so to speak, and some of those which are activated as well, both in the periphery and also in the parenchyma of the brain and spinal cord. And each of these receptors is stimulated by a ligand called Gas6. And there's a second ligand, which is similar, but which only stimulates MERTK and TYRO3. Now, these particular receptors have already been shown to be really important for other autoimmune diseases, in particular systemic lupus erythematosus. And they have three particular functions. The first is that stimulation of the TAM receptors can keep the target tissue cells alive. And we know that it's a survival factor for oligodendrocytes, for example, the myelinating cells, which I mentioned before. The second is that when the TAM receptors are activated, they co-localize with other inflammatory receptors, in particular the interferon genes, and they modulate the activity of these interferon genes from being pro-inflammatory to anti-inflammatory. And the third really important function within the brain, as we shall see in a second, is that the ligands, Gas6 and protein S, can bridge between the receptors expressed on the innate immune cells and particular proteins expressed on dying cells. And that bridging stimulates the immune cells to phagocytose, or actually act as a vacuum cleaner for the dying cells. And why is that important? Well, it's important because it actually cleans up the microenvironment to make it receptive to repair or remyelination. But in addition, it prevents the presentation of further molecules or self-antigens on the HLA molecules, and therefore prevents the acceleration of the disease with time. And the importance of these immune cells or phagocytosing cells was shown some years ago by one of our colleagues, Robin Franklin, in which he took an animal model of disease, and these animals normally spontaneously remyelinate, as seen with these black lines around the light-colored axons. But when you deplete the animal of these immune cells, that remyelination does not occur. You don't see any black lines around the axon here in the reparative phase, indicating that the immune cells are absolutely critical to the remyelination process. And this is leading to us to a more sophisticated perspective now of what drives autoimmune diseases like multiple sclerosis. We have the adaptive immune cells, the lymphocytes in the periphery, which, yes, they target the brain. But in addition, the innate immune cells, the cells which can phagocytose and which are responsible for presenting the antigen, a yin and yang, so to speak, their activity is really important in modulating what occurs. And the target tissue is also not passive. It's not just the brain being targeted in isolation. The response of the neural cells within the brain is ultimately also important in determining what the long-term outcome might be. So given this background, we became really interested in understanding how the TAM receptors functionally might be involved in modulating a disease such as multiple sclerosis. So the first thing we did was to take knockout mice, transgenic mice, in which the Gas6 ligand, which normally stimulates the TAM receptors, is depleted. And we subjected those and wild type or otherwise normal mice to demyelination. And what we found was that in the absence of Gas6, there was a much, much more robust immune response than in the otherwise genetically normal mice. And when we took the insult away and looked at the repair of these mice, we saw also that the Gas6 deficient mice had much less in the way of remyelination than the normal mice, as seen here in this electron microscopy. We also wanted to take this further to look specifically at what the MERTK gene, that susceptibility and severity gene, might be doing in this mix. And to do that, we generated transgenic animals in which we so-called floxed critical areas of the MERTK gene. And these animals in isolation function normally, but when you breed them with another transgenic mouse in which there's a particular protein expressed in particular immune cells, you can use these flox sites to actually delete that part of the MERTK gene and therefore selectively delete MERTK from that subset of cells in the body. So MERTK is normally expressed elsewhere, but it's deficient in the immune cells of interest. And what we did was initially to delete the MERTK gene in the cells expressing the HLA gene, which normally expresses or it normally presents the self-antigen to the lymphocytes. So we wanted to know whether MERTK normally acted as a brake to the autoimmune process and whether the disease might be actually accelerated if MERTK weren't expressed on these cells. So what we did then was to induce a particular type of demyelination called experimental autoimmune encephalomyelitis by injecting this antigen, a myelin antigen, into these animals. And these animals stereotypically develop a disease with hind limb paresis within about 15 days of the inoculum. And what you can see here is looking at the clinical profiling of normal mice, which have received this inoculum, as opposed to the MERTK deficient mice. And what you can see with this clinical grade score on the left, with 3 being frank paraparesis, is that the MERTK deficient mice are developing more severe disease earlier, indicating that indeed MERTK appears to be acting as an accelerator. And interestingly, this effect was mainly seen in male mice as opposed to female mice. And that really tweaked our interest because although I've told you that the disease is more common in women, it's actually more severe in general in men. And this has really given us some interesting data to suggest that there may be some important points here in relation to the sexual dimorphism, which is occurring in relation to multiple sclerosis. We've also deleted MERTK from innate immune cells within the brain, which are normally undertaking the phagocytosis to keep the myelin clean, so to speak. And what we've already identified is that there is a myelin abnormality which develops in these mice in the absence of MERTK expressed by these phagocytic cells, indicating that there is a nexus or an interaction between the innate immune system and the ability to lay down appropriate myelin in these animals. And when we looked at the phagocytic potential of the MERTK deficient animals, we see it was compromised in comparison with wild type animals. And when we actually looked at the wild type immune cells and added in Gas6, we saw that there was much, much more efficient uptake of the myelin debris seen here in green, as opposed to the absence of Gas6, indicating that Gas6 was a very important accelerator of this phenomenon. So all this is very well. This is what we see in mice. How can we translate this ultimately into the clinic? In the last few minutes, I just want to share with you some of the work which we're doing in a dedicated way to do this. And I want to concentrate on three things. First, the expression profiling of MERTK in the MS brain. Secondly, our work to develop a radioligand to assess MERTK expression in the central nervous system on the back of some knowledge about the expression profiling in pathological tissue. And thirdly, just to mention briefly one of several therapeutic approaches that we're adopting. Now, our colleagues at the Cleveland Clinic some time ago had shown that in areas of demyelination, there was actually disparate expression of MERTK from patient to patient in the area surrounding the focus of demyelination. And you can see a patient here with high levels of MERTK expression around the plaque, whereas this person had basically very little expression whatsoever. And we've gone on from this to quantitate the degree of expression of MERTK in different plaques in different people. And we found that the variance in expression profiling is probably about five-fold. And so we now posit as to whether there may be a different clinical course for those who are actually low expressors as opposed to high expressors, given the preclinical data that I've already shown. And so we really want to develop techniques which allow us to actually measure that level of expression in different individuals in real time to be able to make these important calls. We already know that this sort of imaging called PET imaging using radioligands has become important in neurology in identifying epileptic nodes or foci in people with epilepsy, and also identifying abnormal molecules, in particular what we call A-beta in the context of people susceptible to Alzheimer's disease. And we're of the view that these sort of techniques are also going to be very important in diseases such as multiple sclerosis. So in combination with Jonathan Bale at the Monash Institute of Pharmaceutical Sciences, we've developed a program to actually develop a radioligand which will actually bind to MERTK and allow us to undertake these PET studies to quantitate MERTK in the human brain. And so we've developed candidates which have selective expression for MERTK. We've radio-labeled those molecules with F18, which is a radio ligand, or an element which has a radioactivity which is safe, which can be bound to a ligand, and then undertaken autoradiography in animals with demyelination to show that this is specifically binding upregulated MERTK in these animals. And so these are demyelinated mice in which we've actually placed on these sections the radioligand, and it's specifically binding to areas of the brain which are demyelinated and expressed MERTK. And when we actually wash out with cold ligand, we see specific loss of that. So we're now at an advanced stage in having a radioligand which is working, and our next steps are to actually test the efficacy and utility of this in vivo. And finally, I just wanted to mention one area we're heading in terms of therapeutics in relation to the TAM receptors. Gas6 is obviously an important molecule in terms of modulating TAM receptor activity, but it's a large protein and it's got a short half-life. So it's not that useful as a therapeutic because it's would have to be used multiple times in a given day. And in addition, it was not going to penetrate the blood-brain barrier. So what we and our colleagues at MIPS have been doing has been modifying this molecule to increase its half-life with a process called pegylation, which has already been used in a number of pharmaceutical agents. Now, in neurology already, there's precedent to actually deliver therapeutics via lumbar puncture to the spinal fluid in circumstances where there's a long half-life of agents. And we're seeing this in some pediatric conditions, in motor neuron type presentations, and also being used in areas such as Huntington's disease in clinical trial at the moment. And our vision here is, once we've successfully increased the half-life of Gas6, is for patients with progressive multiple sclerosis to be able to contemplate giving this long-lasting Gas6 intermittently, perhaps every few months, via the spinal theca to inhibit the neurodegeneration which would otherwise occur, which we believe activation of the MERTK protein and related TAM receptors might be important in inhibiting. So what emerging insights have I hopefully given you today? The first is that we really now do have a better understanding of who gets multiple sclerosis and why. And we've identified some key environmental factors which are relevant and which we can potentially modulate. And in terms of the genetics, we're working hard to try and identify polygenic scores which might be of some use in buttressing and further strengthening that perspective about susceptibility based on the environmental data. We are also actively involved in trying to intervene at the first clinical event, and that represents the result of the PREVAN study, and we'll have that result hopefully within the next few months, and hopefully that will be positive. We have some ability to identify prognosis in newly diagnosed patients, and using techniques such as MRI, we can detect disease evolution, but we need to have more sophisticated techniques to actually extend this to the molecular level. And the PET imaging I've explained is one example that we would think may have a role in the future. And we're working hard to develop techniques to impede neurodegeneration. Targeting the TAM receptors, as I've indicated, is one particular approach in that regard. We're also working hard on ways to repair the damaged nervous system, but as you can imagine, that represents a very hard ask. And the first thing we should be doing is trying to inhibit damage before it occurs. And that's a very strong focus for us. So in summary, I think you'll appreciate MS is complex. The focus for us is shifting from an inflammation-only approach to now actively consideration of a neurodegeneration and how to intervene and inhibit neurodegeneration. And the key for us is to prevent long-term disability. So without further ado, I'd just like to thank members of my team who are involved in this research listed here on the left. Our great collaborators, both locally, interstate and internationally, of course, the Florey, and also key partners, the University of Melbourne and the Royal Melbourne Hospital, and of course, also our funders. Thank you very much for your attention today.
So we have some excellent questions that have come through. So Trevor, the first one is from Elizabeth, who asks, she says her daughter, November born, has multiple sclerosis. And both of she and her daughter had extreme forms of glandular fever as young people. She notes a lot of monocytes in one of your diagrams. What is the relationship between this season of birth, glandular fever, and multiple sclerosis?
Right. The important point is that most of us are exposed to the Epstein-Barr virus, but in previous times, probably that occurred quite early. And the hygiene hypothesis has for us that the exposures to many infectious diseases, including EBV virus, may now be delayed, and that our immune systems are different at this point of time, and that there's a difference in terms of our response. And the cells which are in particular susceptible to EBV are what are called B cells. And these are B lymphocytes. And I didn't mention it, but in many instances of chronic MS, there is evidence of B cells in so-called pseudogerminal centers within the meninges of people with multiple sclerosis. And it's controversial, but there's been some suggestion that there is a propensity of EBV in these lymphocytes. So this has led to the assumption that EBV may be a key driver of the pathogenesis of the disease. And there are some trials, actually, in particular from a colleague, Michael Pender, in Queensland, who's exploring this possibility with antivirals to see whether there may be a possibility to reduce virus load, so to speak. Now, that's a heroic trial. It's very provocative. We're all looking for the results of that trial with interest.
Right, thanks. And a related question is, how does the late exposure to Epstein-Barr virus correlate with suggestions of increased risk areas, such as the Orkney Islands?
Look, we still don't know what is driving some of these clusters. The Faroe Islands was another good example of it. It was felt that the British military might have taken something into the Faroe Islands, during the Second World War, which changed susceptibility. The honest truth is that we don't still know what are the responsible factors in those areas. And there are two sort of complementary views about where we are with regards to susceptibility or environmental susceptibilities. One relates to the field effect, and the second relates to exposures which are relevant to nuclear families. And there's a trade-off between the two, basically, to some extent. We also recognize that in some instances, there may even be exposures in utero, which are important, which are hard to interrogate, but nevertheless may be very relevant. And we see that in relation to changes in risk according to season of birth. And the idea there is that maternal exposure to UV during various seasons differs, and that they may lay down a certain susceptibility in the fetus according to which month of gestation they're at which season of the year, basically.
Yeah, and it's very interesting in this time of COVID to hear there's quite a lot of interest in the interplay between the COVID infection and vitamin D, isn't there? I know that's a very active area of work in Europe at the moment. And then we have two other questions related to vitamin D. Two people have asked, are there any preliminary results from the Prevance trial? And one person has asked, what's your opinion of vitamin D supplementation at the moment?
Yeah, we don't have results, and we've been very rigorous at the moment in making sure that we don't confound the trial. And look, it's been a journey, and I think that it's been a very well conducted study, and I think will definitively answer some important questions. Anne Louise and I were involved in many discussions actually at the onset of the trial as to whether it should include a placebo group, the dummy tablet, so to speak. And we felt that this was really, really important to do, because the honest answer is that although we know there's a correlation with vitamin D deficiency, we didn't know that it was causal. And so we don't yet have clear-cut evidence to actually say you should be taking vitamin D supplementation at the time of a first demyelinating event. And hopefully very, very quickly, very soon, we'll be able to answer that question one way or the other. In terms of clinical practice, I personally think it's very reasonable to know what your vitamin D level is. And I think it's reasonable for it to be in inverted commas, a normal range. And that's important for other aspects of health, bone health, et cetera, as well. I've also seen one or two instances where people have undertaken heroic measures in terms of trying to increase their vitamin D levels to extraordinary levels. And they've taken calcium supplementation themselves as well, in addition to the vitamin D, and have ended up in significant health problems related to that. So I think that one has to be careful in an evangelical approach to this at this point of time. I think a halfway house approach, waiting for the results of the vitamin D trial, the Prevance trial, is the appropriate way forward.
Yes, and I think that's a lovely moderate approach that, certainly people with MS shouldn't be vitamin D deficient and aiming for normal range. So Trevor, then we have, we're getting low on time, but we have a question about a focus on physio. Can exercise assist in remyelination?
Once again, a really good question. And I think that we know that exercise can assist. And it's a question of how much it can assist. And I think it assists to a variable extent in different people. I think that there's a compensatory perspective, there's a recovery perspective, there's a maintenance perspective. And then, as you're implying, the $64 million question is there an activity dependency to remyelination? And in animal studies, we believe that there truly is an activity dependency to that. And that's been via a variety of techniques, whether it's been with what we call transmagnetic stimulation or by really sophisticated techniques called optogenetics. There are some boutique clinical trials at the moment which are combining TMS with physiotherapy in an attempt to see whether there can be greater improvement which occurs in that context. I'm a strong advocate of physiotherapy and at least maintenance physiotherapy for a significant percentage of people with multiple sclerosis, because I think it helps to optimize function. And it also is an important perspective in terms of mental health as well, indirectly, for people to think that they're not just victims of a disease, that there are things that they can actively do to modulate the outcome. And I think that's really important. I also have some anecdotes as well, which are really instructive, perhaps. And I can remember a senior executive who was basically almost wheelchair bound. They decided to retire. They took on an active exercise program, and 15 years later, they walk in to my consulting room, perhaps not in the last few months with COVID, but it's been just so instructive to see how the change of lifestyle for that person was such a meaningful thing. Now, I'm not advocating that or suggesting that that's going to be the case for everybody at all, but I think that there is a clear-cut role for physiotherapy to a greater or lesser extent for the majority of people.
Okay. And there still are about 4 or 5 questions, but we're coming a little out of time. I'll come to one that I think is related to COVID that I think a lot of people are interested in, and that is, because MS patients have an overactive immune response, does this mean they'll have more severe reactions to COVID-19 or have problems with the vaccine against COVID?
This is a really good question, and there are many facets to it. I choose to think that, yes, the immune system is active, but in targeted ways. The targeting, unfortunately, is to the brain and spinal cord. But in general, it's restricted in that way. We don't generally have people with MS having inappropriate autoimmunity targeted to other organs. There may be a slight increase, but it's not great. So that's from first principles. And the experience in terms of what has happened in Europe, in particular in Italy and France, unfortunately in those dreadful months between March and May, is that they didn't see people with MS having worse outcome just because of MS, basically, in terms of either susceptibility or in terms of negative outcome. What was seen I'll just finish this particular point, I think it's an important point, is that the risks for MS were the same as the general community. That is, that it was with age, and it was with disability, basically, that there was some emergent risks related to the COVID, basically. So the diagnosis of MS itself, per se, we don't believe provides a risk. It's what follows in terms of aging and disability related to aging, which may provide some risk. And the final thing is in terms of therapy, the data to date show no compelling perspective about increased risk related to the therapies. The only one potential wobble, which is still a maybe, is the perspective of ocrelizumab, which targets the B cells.
Thank you. So please join me in thanking Trevor for his fascinating perspective on progress in multiple sclerosis. A special thank you to all of those living with MS who've participated in research over time to contribute to these advances in prevention and treatment. Finally, please don't forget to email or call our fundraising team if you have any questions or would like to donate to our research. We look forward to keeping you informed about Florey research.